Thank you, Jim. Thank you all for your attention and for inviting me here. It's a pleasure and an honor to be here with you. It's also a challenge because, in fact, the prior speakers have been both numerous and superbly expert. And so they have said many of the key and most important things to say. And in fact, much of what I say will be iterative and will hopefully reinforce their points. I think that we really have two very big issues here before us with neurocognition and obesity. One is a more general issue. How do we do research well? And then there's an issue that relates to a specific point that I think is very much on people's minds, and that is about addiction. What is it? And I'm going to address the former a little bit more. That is, how do we do things well, and within that try to highlight a few specific areas to get us thinking. But I think the addiction thing I will spend a moment here to talk about without showing any slides about it, because I think it's really a vital piece of the agenda. And I particularly like some of the talks trying to go through it and think about not just showing you data, but saying which data would in fact be relevant. And I think this is a relatively new level of sophistication we're having in the conversation about the possibility of food addiction to begin asking that question. If we want to know whether X exists, we first have to know what X is. If we want to know if something isn't X, we first have to know what are the criteria for being an X. And I think we've heard more sophisticated dialogue about that today than previously. And I think that Dr. Avina's question about, to one of the other speakers of saying, well, if you're not sure about the DSM, where does that leave me? What do I do? And this is kind of a stuck position because, in fact, without having a set of criteria, we can't ask if those criteria are met. So we've got to have a set of criteria, but we'd be foolish to simply reify the DSM and say, somebody wrote it down, therefore it must be right, or therefore it must be good. And this reminds me very much of a conversation we had on a slightly similar topic a few years back, which was, is obesity a disease? A very similar sticky question. How do you answer it? And I actually wrote 2 papers about that. In the first paper I wrote, we said, no, we don't think it's a disease. And that wasn't a popular view. And we actually revisited it, and we talked about it some more, we had lots of debates. And I actually came to change my view because I felt not that other people who said it is a disease were right, but in fact that we were all answering a non-question. Simply because we can pose or phrase the question doesn't mean it's correct. As Neil deGrasse Tyson once taught me, he said, just because I can ask you at which temperature does the number 7 melt, it's a perfectly grammatically correct question, doesn't mean it is a meaningful question that actually permits an answer. And so similarly, the question of is obesity a disease predisposes that you actually have a definition of a disease that works, that you can empirically compare obesity to. Well, it turns out when you really study this, there really doesn't seem to be very clear definitions of diseases that admit all the things you want to admit and have those be called diseases, and that don't admit all the things that you clearly say, well, we don't want that to be a disease. So you can use some definitions. Of course, obesity is disease by evidence. It's a condition of the body. It leads to impairment of health or shorter lifespan. It has genetic or physical or social causes, which is one definition you get. I say, well, being a man satisfies all those criteria. It's a condition of the body. It's associated with shorter life and other diseases and has genetic causes and social relations and so on. So clearly that definition doesn't work even though it's written in things like Webster. And so just because the definition is written in DSM doesn't mean we necessarily need to adopt it for scientific purposes. Remember, the DSM is a diagnostic manual. It is a manual for diagnosing individual patients as having things they can be reimbursed for when receiving psychological and psychiatric treatment. It is not a tool for defining whether or not things, one thing causes another, or whether categories of things exist and so on. So I just want to point that out, and I think that level of conversation, say, can we get to a definition of addiction that in fact is scientifically meaningful, that admits a key test, so that then you can say, aha, with this definition in hand, I may now say, what would be the experiment that I could do, even if I can't even do it in practice, but at least in theory, what is the experiment I could articulate such that at the end of that experiment, I will be able to tell you, food is an addiction, food is not an addiction. Only with a definition like that can we get to that point. If it turns out that we can't get a definition like that, it may be that, very much like in the case of is obesity disease, we came down and we said, that's an ill-posed question. It's not is obesity disease. Really the question is, do we wish to call obesity disease? That we felt that what becomes a disease is a negotiated process. It is not an empirical scientific process. And it may be that perhaps that's what we'll get with addiction of foods. We may say, it's not really a scientific question. It's a negotiated process. We say, is it useful to call foods or certain foods addictive, as opposed to can we prove it or disprove it? Anyway, so that's a few thoughts I just wanted to give on addiction because I was really very impressed by the high level of thinking on that. Now I'm going to talk more about what I had planned here, which is really more some methodologic pieces. This is my disclosure. I won't read it to you. My email is up there. If anybody wants my slides, just email me. I do want to acknowledge Andrew Brown and Ed Archer, who were very helpful in helping me think through this talk and also preparing some of the slides. This is an outline of what I'll go through. Talk about the idea of being blinded by the brain, like the Springsteen song, Blinded by the Lights. Challenges in conducting neurocognitive research. Do we interpret studies well? Do we ask questions well? Conclusions and recommendations. That is, I don't think there's a question of should we be studying neuroscience in obesity. Question is, given that we're going to study neuroscience, how do we do it well? So blinded by the light of new technologies in this case. And I want to point out that this phenomena is, I think, not unique to neuroscience and obesity or neuroscience of food intake. That is, in my experience, this happens whenever a new technology is put into existence. I don't know that it happened, but I can't help but suspect that when Galileo invented the first or developed the first really good telescope, that maybe there were exaggerated claims about what telescopes could do, and people got confused about that as well. We see this in many areas. In 1999 or so, I began to see this with microarrays for gene expression. And people would come up with these amazing heat maps, and they'd look at this and they'd draw conclusions, and you would see audiences as big or bigger than this one of rapt, senior, solid scientists who had learned statistics and knew how to ask hard questions, just with their mouths agape, looking at colored 3D graphs as though there was somehow something they could discern from this. And you say, oh, is there a p-value, a confidence interval? Is there a hypothesis being tested? Is there some indicator of stability? But no, it's a pretty colored graph. And we published then a paper in Nature Genetics on this, on the idea of sound epistemologic foundations. And so you may want to take a look at that if you're interested in microarrays. And actually some of the ideas may even apply to some of the areas in neuroscience. We see it with big data. Big data is hot now. We see all kinds of wonderful claims. And Google can predict flu outbreaks better than the CDC. Maybe. Some evidence says, gee, their model doesn't hold up so well. And here's a nice piece adapted, I think, from originally some work by Nassim Taleb on the foibles of big data. So all of these things become faddish. We can't leave our brain out. All the same principles of good experimental design don't change when a new technology is introduced. So why do I say that? Well, think about brain images. This is, you all remember the TV commercial, this is your brain on drugs. Well, let's think about a few other analogies. This is your brain on music. So this was one of the studies that was put out showing fMRI scans of looking at different music effects. And then this nice study came out by McCabe and Castle. It was a very clever article. So it included 3 randomized controlled experiments in which what they did is they gave the same papers to investigators and asked them to rate aspects of the convincingness and clarity and quality of the papers. But they did it with and without images of brain scans. Right? So the data are the same, the findings are the same, everything's the same, except there are or aren't pictures of brains, quote unquote, lit up. Right? There are no lights in the brain, but we all hear that terminology. And here's what you see for ratings of the statement. The scientific reasoning in the article made sense. So on the left, you have the control figure, no brain scan, and in the middle, you have a bar graph showing the data, and on the far right, you have a brain image. And suddenly, because the brain image is there, even though the same data, the same experimental design, suddenly it's more convincing. So that means we need to take a deep breath, right? Doesn't mean we shouldn't put brain images in, but we need to take a deep breath as scientists and say, hold on a second here. Would I be convinced of this statement if it was just a bar graph? And if not, why does this picture somehow make it seem like there's something more here? Is that legitimate? In their second study, they did a sort of a formal mapping of the brain shown on the left versus the image shown on the right. Again, same data, and ratings of the statement, the scientific reasoning or sound, go up with the brain. And this one was a much smaller effect. And this one was, are you convinced that the conclusions and title are appropriate? And again, with and without the brain, and there was small increases in the extent to which people saw these statements as appropriate. So clearly we are dazzled. We are dazzled by the razzle-dazzle of these beautiful color images from the brain. Doesn't mean we shouldn't look at them, but we need to take a deep breath and not get addicted to brain images. Okay. Next question is, do we answer the question at hand? And somebody previously showed a very nice slide of this, so I won't recapitulate the whole thing, of looking at peeling an artificial apple versus peeling a real apple in or out of an MRI. We need to keep in mind that looking at a picture of food is not the same as eating a food. Eating a food in an MRI is not the same as eating it out of an MRI. Peeling an artificial apple is not the same as peeling a real apple, and so on. And so we always have to just ask, are these questions pertinent? So it's an obvious thing, but again, we can't forget that because we're so excited about the technology, to not ask about the fundamental aspects of the thing we're studying. This is a very famous paper. Some of you may know it. This is the post-mortal ichthyological cognition. That is a dead salmon thinking. So what was done is they took a dead salmon and they put it in an MRI. And they did it with, two different conditions, and then they, saw which parts of the brain lit up, and you see some parts lighting up. And clearly this is nonsense. I don't think we don't know about live salmon, but dead salmon, I think we're pretty sure are not thinking. And what's going on there? Well, there's no correction for multiple comparisons. Someone else said, well, how about a mannequin? So they hooked up a mannequin to some brain scanning technology, and they did it at rest and giving the mannequin a task, some things on a computer to look at. And then, what they got is statistically significant differences in mannequin, brain lighting up when no adjustments were made for multiple comparisons, right? The more you look, the more you see. And so if you do enough statistical significance tests, you find things. And there's the mannequin brain and so on. All right, now, these are uncorrected, right? If you correct it with Bonferroni correction, that all goes away. Now you would say, oh, come on, these are just silly examples. Certainly valid fMRI researchers, professionals, are not going to do anything so silly. They're going to know to use multiple comparison corrections, and only the corrected results will be publicized. So this is your brain on sugar. This is from a JAMA paper, and you can see some different areas lit up under different things, got in JAMA. And this is a description of it, effects of fructose versus glucose on regional brain cerebral flow, blah, blah, blah. When whole brain contrast maps were directly compared between fructose and glucose sessions, fructose ingestion produced greater activation in the hypothalamus and striatum. And you can bet the media had a field day with this one, all right? But keep in mind, these regions did not survive whole brain correction analysis. So you can look at this a lot of different ways. One is that, maybe this shouldn't have been published, or maybe it didn't deserve the media interpretation it got. On the positive side, we often hear young people say, part of the problem with publication bias is I can't get my paper published if I have no significant results. And it's clear you can actually publish it in JAMA if you have no significant results. So maybe it's good. What about multiple comparisons? Did I pick off an isolated example? Is that the rarity, some freak paper that just got in JAMA? Well, here's from 2008 journals, number of papers looked at. 60% in social and cognitive affective neuroscience did multiple comparisons, and only about 62% in the Journal of Cognitive Neuroscience did. And you see other sites here. And so what we see is that 25–40% of papers are not including any correction for multiple comparisons. Well, if you look at every voxel and every pixel, of course you're going to find significant things by chance. How do we then interpret these? Here are some headlines. Skipping breakfast to lose weight makes you fatter and far more likely to raid the vending machine. This is actually based on a presentation at a proceedings, MRI results, and observations of how much subjects ate at lunch after skipping breakfast. No measures of body weight were included, no vending machines were included. And so there's this huge extrapolation. Here's two headlines from other studies. Eating cheese and meat may boost self-control. Official, chocolate stops you from being grumpy. The study was a study of acute tryptophan depletion modulating reactions to an ultimatum game. So tryptophan is contained in some of those foods, but it wasn't those foods. Headline, Neuro boosts minds, moods, and more, a drink review. And if you actually go, there's no study. It's a blogger reading the names of drinks that a company sent her. Now, you might say, well, this is just whoever writes the headlines. Why are you telling us? We're scientists about this. This is not our problem. We can't be responsible for what the media do with our study. Well, here's one that just came out last week. This is not about food intake, but this is about concussions. And this journalist who sort of dug a little deeper into the study had a concern and writes about the press release that fell and hit its head. Basically pointed out that concussions, like addictions, like obesity, are hot topics now, and they get great media attention. And what happened when this paper came out was that people kept talking about the effects of concussions based upon these brain studies. And then this particular journalist, unlike most journalists, went back to the original paper and said, wow, I can't find the word concussion appearing a single time in the original paper. But then went to the press release that came from the author's institution, and the word concussion was in the title. So she contacted the author and said, well, what's going on here? And the author said, well, I kind of thought people would understand concussion, and, I did wonder about it, but I decided we would go with it. So clearly, we are complicit in this, we academics at times, and this is a good example of that. Andrew Brown showed this yesterday. What you see on the far left is abstracts in journals, and this is from an analysis by this group by Yavshitz et al. 17 of a number of abstracts looked at, the darker blue on the top, contained what Yavshitz et al. Called spin, meaning they sort of took a result and tried to spin it to make it seem like more than it was, and they had a coding system for that. 24 did not include spin. Now you go to the press releases. If the authors include spin in the abstract of the paper, then the press release almost always includes spin. If the authors don't include it in the abstract, the press release rarely, but sometimes includes spin. And as you go to the news pieces, if the press release is spun, then the news is spun. And so I think we really have to recognize that we in academia are complicit in creating these exaggerated claims and these misinterpretations, and so we have to do better with that. The media, of course, will eat it up. And this just shows what does the media cover. This is from a paper that came out this year. And I won't go through everything, but basically what you see is that the weaker studies are the ones that are most often selected. So for example, time series, which we would generally consider very weak evidence, ecological correlations over time, are picked up far more often than are properly randomized controlled trials. Right? Because time series can be more exaggerated. Because I can put anything on an x-axis changing with time and find something that changes on a y-axis and vice versa. They're very exaggerated, they're very exciting. We need to be careful, we need to not feed the media on this. All right, so that's about interpretations. Do we ask scientific questions well? Many, many papers are out there in the philosophy of science literature about cognitive science in general and about brain scans in particular. And I encourage you to take a look at some, and I've just cited two that I could quickly find. And here's a quotation from the second one I'm citing. It says, neuroimaging uses indirect methods to generate data about surrogate parameters for mental processes. This leads to an interdependence between hypotheses and data. Due to their inferential proximity, neuroimages, blah, 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 blah. Well, this is actually a really good point, and I really admire philosophy philosophers of science for their rigor of thinking. Unfortunately, I wish they could write in a way that more of us could actually understand what they were saying. Someone who's really good at writing in a way that other people can understand is David Katz from Yale, and he's basically, I think, making the same point here, but in language that probably more of us can understand. And what he talks about is a particular paper by Gerhardt and colleagues in Archives of Psychiatry, which showed that individuals scoring higher on a scale for food addiction have more intense responses in the reward circuitry of the brain revealed using fMRI scans than people who are less food addicted by this scale. The media attention to the study has generated what Dr. Katz would suggest that it delivers an epiphany. And he goes on to say, but I think the case is just as strong that it delivers a tautology. Those who report receiving a stronger, more irresistible reward message from their brain in response to food have a stronger, more irresistible reward response in their brain in response to food. In other words, what did we learn from the brain imaging in this particular study that wasn't really there already? And so saying we've identified people who find food very rewarding, and then we say in response to food, the reward centers of their brain seem to indicate reward. It's not clear that we actually learned something from this. And so we need to really say with many of these studies, what's actually being learned here? And again, if it wasn't for the excitement of a brain scan, does this really tell us something new? I think we need to be aware of the potential for reification. That's a fancy word for saying that we think what we name something is what it really is, making something real. Reification and equating our names for things with the things themselves. And this actually, I think, was touched on by one of the prior speakers. Consider the following. So this is a real scale that exists. It's called the Magical Beliefs About Food and Health Scale. It contains questions such as, if we don't somehow clean our bodies of unhealthy toxins will remain in them. The scale has been checked for psychometric reliability. It is reliable. It has been correlated with other things, which indicates validity as a measure of something. In other words, if something correlates with something else, that's prima facie evidence. It must be measuring something. But the question is, what is it measuring? And I'm not criticizing the scale. I do think it's measuring something. I think it's measuring a way of thinking. I don't think it's measuring magic. We have the Yale Food Addiction Scale. It's called the Food Addiction Scale. It contains questions like, I find that when starting eating certain foods, I end up eating much more than planned. It has been evaluated for psychometric reliability, just like the scale of magical thinking about food. It is reliable. It too correlates with other things, which means it's measuring something. The question is, what is it measuring? And just because it's reliable and correlates with things, the magical belief scale doesn't necessarily measure magic. The Yale Food Addiction Scale, just because the authors chose to call it the Food Addiction Scale, doesn't necessarily mean it measures food addiction. It may, I'm not saying it doesn't, but that is not ipso facto evidence that it does. And we need to be careful about getting too drawn in to the idea that because someone else named it, suddenly it's real. So in conclusion, I think that neuroscience research, including brain imaging studies, can be fascinating and cool. To also be valuable and valid, we need greater efforts to be sure that we are asking meaningful questions with valid designs, analyzing the data properly, and interpreting the results fairly. New technologies may seem magical, but are not magic and do not change the fundamental logic and statistics of experimental design. And at this point, I know that many of these things are challenging issues. They require a lot of thought. So I hope that some of you would like to come visit us in Alabama. We'll take you on a canoe ride, and we can talk about it some more in the canoe. Thank you.